Hello and welcome. It's time for asynchronous recorded lecture 31 for Bio 145 Microbiology. This lecture is scheduled for Friday, April 10th, 2026. Our topic today is respiratory tract viral diseases. It is a rather long lecture, but it has very valuable and practical information. So enjoy. So this kind of reviews some of the spots in a viral life cycle where these drugs work. So uncoating, we talked about those. Nucleic acid synthesis, we have the nucleoside analogs, the ones that look like nucleotides. And then I kind of said the ones that don't look like them. We talked a little about protease inhibitors. Okay. And we'll talk a little about this a little later. We will get here, but just a little later than neuraminidase inhibitors. All right. So you can say, if you kind of know the pathway of how viruses reproduce, those 5 steps, understanding where the drugs work, okay, is not that hard. Okay. So my question here is this. Dr. Crystal Clear Thinker has developed an antiviral compound when applied to a host cell that's infected with an envelope sense virus, okay? Sense RNA virus. Think about that. The drug crosses the creation of shortened viral RNAs. The compound is, so you can stop right now for yourselves. Stop, think about it, but I'll just keep going on. Number one, and is it an interferon? Okay, so thinking back, or is it an uncoating, or what is it? We'll think about shortened viral RNAs. All right, so number one, interferons, those lead to proteins not being made. All right, so it's going to stop viral proteins. It's not going to shorten the RNA, so it's not that. An uncoating inhibitor, that means that it doesn't even get, remember, the virus doesn't even get out of the endosome. So if they can't even make any viral RNA, so it's not that. A reverse transcriptase inhibitor. So you think, well, you inhibit reverse transcriptases, that would give you shortened DNA, right? I can't be. Plus, it says envelope sense RNA virus. Well, that's another reason. No, reverse transcriptase is only for retroviruses. So that's 2 against that one. So it's got to be a base analog, and it makes sense. Yeah, because beta analogs lead to shortened genomes. So if you have an envelope sense RNA virus, it'd have a sense RNA genome, so it would shorten it. Remember, it's a dead end. It's a dead end. It's a mimic. So the answer is D. I hope that makes sense. So enough of the intro. Let's start applying this knowledge to the thrilling world of respiratory viral diseases. And the first one we're going to talk about is the flu. Okay? So think of viral infections of the cells in the human respiratory tract. And you want to think of the flu as a virus that's going to attack actually the upper cells, all right? Think your throat, okay? Stuff like that. And also think lower. Think those ciliated epithelial cells in the lower tract too, okay? Among other cell types, okay? But just think it's both. A little cocktail. In the United States, every year, about 40,000 people die due to the flu or a secondary disease triggered by the flu. Okay, you might have heard about this. They've been talking a lot when they've talked about COVID being a pandemic and how bad a pandemic can be. Well, back in 1918, there was a flu pandemic known as the Spanish flu. It spread throughout the world. And again, remember back then, the understanding of microbiology, treatment, of disease, vaccines, all that stuff wasn't developed as it was now. And they estimate over 40 million people died because of that. So flu can be really bad. The agent for the flu is, we just call it the influenza virus or influenza virion. It has 8 separate antisense RNA helices. So it's got 8 strands of antisense RNA that makes up its genome. So if it has antisense RNA, what must be a soluble protein? Okay, if it's got antisense RNA, it comes in and nothing can happen to it. It needs to be converted to sense, right? So it's got to have what? It's got to have a viral RNA polymerase, right? It's got to bring in its own viral RNA polymerase to use on the antisense RNA to make complementary sense RNA. And also, it's going to have a capsid and it's going to have an envelope. Now, on the envelope, you're going to see these things called spikes. Okay, we've talked about them before, and they just contain proteins. And what are the key ones that you find in the spikes on the envelope of influenza, or just call it flu virus? Okay, here. So let's look at the virus, the little cartoon first. So you've got your 8 antisense RNAs, 1, 2, 3. Yeah, you can count them, 8. Then you have your capsid, which is shown here in yellow. Okay. And then outside in purple, you have your envelope. And sticking out of your envelope are these spikes. So you see the N spike with neuraminidase and the H spike with hemagglutinin. Okay. So hemagglutinin is important because that is the ligand that is coming out of the viral envelope. It is the ligand, hemagglutinin. It's a protein. Okay. And it is going to attach to a receptor, and the receptor is on the host cell. Okay. The receptor is a neuraminic acid or sialic acid, but let's go with the old-fashioned name, neuraminic acid. All right. So let's go another drawing here. We're showing the H, the hemagglutinin right here. It's shown here in this little red stub here. And it is binding to, well, it's labeled glycan, but that is the neuraminic acid. Neuraminic acid. So that's kind of interesting that it's a protein ligand on the flu virus binding a receptor that's not a protein. A receptor is actually a long polysaccharide that's on the human cell. Okay? Neuraminic acid. Now, just to remind you, the H is found coming out of the envelope. Okay. Now, there's a lot of flexibility in play between the H protein, the ligand, and down here, the neuraminic acid, the receptor. There's a lot of flexibility. They don't have to be specific, you know, how they fit each other. All right. So it's kind of it's kind of different than when most people think lock and key. No, it's the key and the lock. They don't fit as tightly, but there's a lot of looseness there, but they still work. So what that means is a big thing. It means it allows for changes in the H structure. So think you can change a few amino acids in hemagglutinin structure without hurting its function. Okay, and that's going to be important later. All right? Because guess what? The H gene, hemagglutinin gene, is going to be very mutagenic. It's going to be mutated a lot, which means the hemagglutinin protein will change a lot in its structure. But it's still, even though if it's changed, it can still fit and work with the neuraminic acid receptor. Okay? Keep that in mind when we talk about things called antigenic variation. That'll be very important. Okay, enough about the H. Let's talk next about the N spike, the neuraminidase. Note, not neuraminic acid, neuraminidase. Okay, what is N? N is not neuraminic acid, it's neuraminidase. It's important for entry and exit of the virus from the host. So it's entry and exit, but mostly people think about exit. All right, so here's neuraminidase. Here we go. Okay, it's also one of the spikes. It's in that envelope, right? And its function, well, neuraminidase, it attacks neuraminic acid. Absolutely, that's what it does. It digests off the neuraminic acid. That's the receptor. Okay, it's on the cell surface. That's the receptor. So that the flu can get away from a sticky plasma membrane. So you're like, huh? What do you mean? Well, remember, H, okay, hemagglutinin binds neurominic acid. And it's a really tight binding. It's like a handshake. So when H binds neurominic acid, okay, here, it's a strong binding. But you don't want it to continue forever, because think about it. This virus, okay, if it's entering, wants to enter the cell. If it's stuck here, it'll never be able to enter. By the same token, imagine this virus is budding off from the cell. Let's think of the other way. It's brand new and it's budding off. And an H seems to scrape by a neurominic acid. They're have such high affinity for each other. They love each other. There's like an animal magnetism there. They will stick to each other just by accident. Okay, even when the virus is leaving. And once they stick, it's like a strong handshake. And it just won't let go. It will not let go. So you're like, okay, how therefore can the virus either go in or leave, or else it's going to be stuck at the surface? Well, like I said, if the H is the ligand and the neuraminidase is the receptor, neuraminidase is like a knife. Okay, it's like a knife. It's going to do cutting. It's going to cut the neurominic acid. Oh my gosh, Jeff. You're not saying cut someone's hand off in a handshake. Would you really be saying that, Jeff? So yes, what I'm saying is neuraminidase is like a knife, and it literally cuts off the quote-unquote hand. It cuts off the neurominic acid. All right, which was sticking to the hemagglutinin. So in this picture, all right, we're noticing they have a baby flu virus budding off right here. It's budding off, and like I said, as it buds off, it just happens that the neurominic acid receptor, all right here, ends up sticking by luck. They just cross against each other, and it grabs onto the hemagglutinin. All right, the hemagglutinin. So you have hemagglutinin bound to the neurominic acid, and now this guy's stuck, the virion, the baby, but can't leave. At this point, the neuraminidase, the N, cleaves that off, cleaves that neurominic acid off. All right, it's doing that. So now this can get away from that sticky plasma membrane and be released. Hope that makes things clear. So H and N are very important. They're used to classify viruses. So for instance, you might hear, oh, this year in the US, there's a lot of type A H2N1 virus. All right, or they'll say type B H3N5 or something like that. So the H and the N are used to classify the viruses. All right, so both H and N, along with doing their jobs in the viral replication cycle, serve as antigens for your host system. All right, they're out there on the envelope, they're sticking out there, they're an obvious target to make antibodies against, if you want to think of it that way. Now, if they change, though, this makes long-term immunity difficult, depending on how much they change. So if the H and N change from one generation to another just a little bit, the memory cells are still pretty decent. But if they change a lot, then those memory cells can become useless. So we want to think again, that can be a chance for antigenic variation. And there really is quite a bit of antigenic variation of H and N from basically between one class and the other. So H2N1, H3N2, the two H's look very different. And also, as I'm going to say, even a virus that's an H3N2, from one generation to the next, the H3 can change a little bit too. It's still an H3, but it's a little different. So when you think of H's and N's, what do you think of them as in terms of general shapes? Okay, so here you've got a picture and you've got some drawing. Supposedly here is the influenza virus. Here's its H1. All right, here's an H1. Here's an H3. Okay, H3. Just worry about H1. Okay, don't worry about H5 right now. I'll explain that one later. And you can say, well, what's an H1? They're both hemagglutinin, they're both ligands, but you can see their overall shape is slightly different if you compare this one and this one. So you can think, okay, the H1s, they're the ones that have like a rectangle look. The H2s have a square look to them. The H3s have an ellipsoid. So let's just say, this one's kind of got the rounded edges. So let's say H3s kind of have an ellipsoid look to them. All right, and so you can kind of differentiate H1s against H3s. All right, and the same thing can be said of the neuraminases. They have different general shapes of N1 versus an N5. Just a little thing when they talk about pandemics of influenza that have gone throughout the world, folks. It's important to think. It's important to think that you can classify them by their general shape, their H and N shapes. You can see them here. They can show what kind of flu virus led to a pandemic, depending on the year. Right. So also, there's three types of influenza virus. So along with the H and the N designation, they'll say they're A, B, or C. A is basically the more virulent group. They have a tendency to infect pretty much birds, pigs, humans, pretty much everything. Wild birds are natural. They're usually the more virulent. B and C are a little less virulent. They don't cause pandemics. They're not as efficient as in entering and exiting the host. Okay, C, it's an obvious why because C actually lacks in neuraminidase. So for C, you have a type C, you have an H1 or H2, but there's no N designation. Okay, so the worst things you worry about are the type A. So if you're type A, H1, N2, that's usually worse than type B, H1, N2. So let's talk about the flu, the mechanism of the illness. Transmission, again, airborne respiratory droplets, either inhaled directly, so someone coughs or sneezes, they have the virus in their mucus. They cough, they sneeze. You either go by them and you breathe it like a vacuum cleaner, or those viruses land and you then touch that surface where they landed, and then you, with your fingers, you rub your eyes, your nose, and therefore you transfer the virus to your respiratory tract. So there's several ways of picking it up. The incubation time is 1 to 2 days, and it's interesting because people will tell you that, man, they were feeling fine at the beginning of the day, but by the end of the day, they were horrible. And how can they have gone down so fast, so badly, so fast? Well, the burst time for a virus, for the flu viruses, are about 6 hours. That's, again, time from entry to when the babies start coming out. That's pretty short. And the burst size, remember, that's how many babies will come out of one infected cell. 100,000. So when you see that 100,000, think, okay, that means 100,000 come out. That means each of them can infect a new cell. So pretty much within 6 hours, each of those one will enter a cell, and then 100,000 will come out from those cells. So you can see 100,000 times 100,000 times 100,000. You can see a lot of your cells are going to be infected and damaged very quickly. The dose is only about 10 to 100, so it doesn't take a lot for the average person to get sick. It's a very, very small dose. If you want to learn more about this in your spare time, you can look at these websites or these YouTube videos. Sometimes students have to do reports in farm school about the flu virus. So if you want to learn more, those are really good resources. I just don't have time to go into more stuff now. The pathogenic mechanism, basically H, binds the receptors of neurominic acid. The N cuts, the virus is endocytosed. All right. The virus then uncoats. All right, because of the H plus being sent into your endosomes. It's a normal response of the body. And like I said, the H going in helps basically everything uncoat, the melting away of the plasma membrane, the envelope, the capsid, so that they all basically melt away. And now you get the release here of genome and soluble proteins. Okay. Once those are inside the cytoplasm of your human host cells, they replicate. It's an antisense RNA virus, so that's going to go through all. It's going to assemble. it's going to bud off, and it's going to spread to new cells. Now, let me just remind you, where the H and N, they're going to be made during the cycle. They're going to be translated. Where they go? Well, if they end up in the envelope, they end up going into the plasma membrane, right? And they form these areas of spikes where the new viruses are going to bud off from, right? So there's going to be points where they're going to bud off from. Don't forget the whole picture there. All right. Well, the infected cells, again, throat, lower respiratory tract, but let's really focus on these ones here. The ones, for instance, in the trachea, bronchioles, those ciliated respiratory cells. These cells don't lyse because it's an envelope virus. They're going to get budded off. They're going to work those cells to death. But after a while, they're going to die. So pretty much you're going to go with cells with a lot of cilia to cells with hardly any cilia at all. So those are the cells of those are important because they're the cells of the mucosal escalator. Remember, those are the ones that are protecting your alveoli. All right? Those are keeping that mucus moving and so you can cough it up. All right? If those get, they're going to lead to secondary diseases, which I'll talk about. Now, the symptoms, while you're going to be fever, really hacking cough because you have that weak mucosal escalator, so it's hard to get that stuff to move, so you have to cough really hard. Body aches, exhaustion. And the virus is shed for about 7 days. So, about the first 2 days, you can be asymptomatic, 1 to 2 days incubation period. But then about 5 days of symptomy, and you are contagious. So that's why they usually tell you have the flu, stay home from work for a few days. Well, how about the fever and the body aches? Where do those come from? Fever is due to what? Release of IL-1. So fever, think the virus swallowed by macrophages. Macrophages are known to produce something in the T helper response. It's something which is also a pyrogen. It is interleukin. Think back, 1. Yeah, interleukin 1. So that helps trigger fever. There's no endotoxin, okay? No endotoxin. Why is it achy? Well, your body produces a lot of cytokines, especially the interferons that we just talked about. And those interferons, those selfless gifts, well, they're good because they help the body slow down viral infection by inhibiting translation of viral proteins. But they are a signal to the body, and they can trigger things like headache, body ache, exhaustion. If someone has the flu, they'll say, I could not get out of bed. The exhaustion of the body were so bad. Well, that's really not the flu making something specifically. It's your body reacting to the flu, especially those interferons, body aches, exhaustion. And later when you see when people use interferons as drugs, that's going to be a side effect. Now, as I said, if your body is fighting it, you often get a secondary infection. The flu, it destroys the mucosal escalator. What bacterium that's just part of your normal flora can now seize upon that, especially in the elderly and the immunocompromised, and lead to what disease? That'll end up, if there's no mucosal escalator, they go to the alveoli, attack the alveoli, go to the blood. Pneumococcal pneumonia. Very good. I'll let you write that in because that's a long word. Pneumococcal pneumonia. Yeah, because that disease often follows a lack or weakness of the mucosal escalator, and that's what the flu will do. Before it can be treated, usually how do you know you have the flu? Well, now there's just what's called RIDDTs, rapid influenza diagnostic tests. They're just immunosticks where they take a little sample of your sputum, mucus, whatever. They dip it, they wait, and they look for the line. And if you have certain flu virus antigens, there's the line, boom. So they can basically diagnose you very quickly in a doctor's office. The treatment, let's say you are diagnosed, you have the flu. And I'm going to say all of these, they have to be given early in your disease progression in order to be effective. If it's too late, there's too many cells infected, it's not worth it. You got to get it early before the virus has infected a lot of cells. One is Zofluza. It came out a couple years ago. It's what it does is basically inhibits the viral RNA production. So it stops the viral RNA from being made. All right, by viral RNA polymerases. It's a very complex mechanism, but it's amazing. And usually it's a little cocktail. It's only one dose. One dose. But again, very early on. The second is amantadine, rimantadine. We've talked about this already. They inhibit the acidification. They keep the endosomes neutral. If there's no acidification, there's going to be no uncoating. However, it's not recommended as much anymore because there's a lot of viruses that are resistant. Why? Well, a lot of usage means selection for resistance. Was there one event that really caused a lot of resistance? Yeah, about 2.6 billion chickens in China were treated with amantadine to stop the spread of flu virus among the chickens. Remember, flu viruses can infect chickens too. So they used it a lot, and because of that, a lot of resistance came, and now it can't be used on humans very much. And finally, there's the neuraminidase inhibitors like Relenza and Tamiflu. All right, and they'll stop the spread if given early. We know about neuraminidase. It's the one that cuts off it cuts off the neuramidic acid. If you inhibit it, okay, here we go. Here's your neuramidic. What happens? Well, the neuramidic hemagglutinin thing stays. It's stuck to the surface, and therefore the viruses aren't released. So neuraminidase inhibitors prevent the release here, and if they can't be released early on, that's viruses are not going to be able to get away from cells and infect new cells. Now, if you look at the data, they'll say, well, if you take neuraminidase inhibitors early enough, such as Relenza and Tamiflu, it won't last as long, maybe 1 to 2 days less, and it'll decrease the severity of the symptoms. And you might think, well, that's really not that big of a deal. For people that are young and healthy like you, it's probably not that big of a deal. But if a person's immunocompromised, elderly, susceptible to pneumococcal pneumonia, that reduction of 1 to 2 days of duration and the reduction of severity of symptoms and signs could be life-saving. So there is a place for those. They're not perfect. Grant, they're not going to cure you of the flu overnight, but they can help you, especially if you are in one of these sensitive groups. So here's a question, part of the recap. Dr. Rainer Schein has treated a flu-infected human cell with an antiviral drug. He waits a little bit. The viruses are captured inside endosomes and end up being degraded in lysosomes. So they didn't get out. The virus didn't get out. So if the virus doesn't get out, it's going to end up going to the lysosome, right, and getting destroyed. So no mature virions ever come out. Thus, he must have used a drug that, here you can pause it and think, but I'll just go into it. Affected neuraminidase function. No, that's not right, because neuraminidase is for entry or exit. This is actually inside the cell, inside the endosomes. Raise the pH. Yeah, that's right, because hey, if you raise the pH in the endosome, there's no acid shower, there's no escape, there's no uncoating. Affected the biosynthesis step. No, this is kind of more the entry step. So it's not biosynthesis because things aren't actually being made for the virus yet. So the answer is B. B is the correct answer. Let's go continuing with treatment and prevention. Usually your immune system, your cell-mediated immunity, your cytotoxic Ts, and antibody immunity, if you give it enough time, they will get rid of the flu. They'll get rid of it. They'll get rid of the virus in your body. Now, one thing I want to make a big deal about is this, is you can rest, you have fluids. Don't give a person aspirin, all right? Especially with a fever, a lot of people want to go aspirin, but aspirin for children can be very dangerous. For some reason that's not clear, this doesn't happen in all kids, but in a decent percentage, kids that if they have the flu and they're given aspirin, they come down with Reye's syndrome. And Reye's syndrome can result in a coma and potentially death. It causes neurological problems. Again, it's not clear why certain amount, certain kids this happens to. I have a personal connection to this. When I was in second or third grade in St. Joachim's Catholic School in Madera, California, a little girl joined the class in the middle of the school year. She was assigned to my group. I remember her name was Kathy. Short blonde hair, liked to play hopscotch. Well, one day she didn't come to school, and that went to several days. And we'd heard, oh, she'd gotten really sick. And then we heard she went to the hospital. And the next thing we knew, we were doing a rosary for her funeral. She ends up dying because of Reye's syndrome. So, if you remember nothing else from this class, do not give a small child aspirin if they have the flu. Please. How do you prevent it? Usually, the common vaccine is a dead agent vaccine. It's given as a shot, it's dead agent. And you can get it yearly, okay? And you've probably heard of the flu shot. So you think about it, what kind of memory cells? Just a reminder, it's dead virus. Yeah, it's memory Bs. Very good, memory Bs. How are they made? Well, it's kind of interesting. Is pretty much the viruses that are going to be in the US in 1 year, it is assumed that they are in Asia the previous year. So basically, the viruses in Asia take a year to get to the US. So our public health experts go to Asia. They figure out the strains that are most common, that are the greatest chance of getting to the US within a year. And they take those and they bring them back, okay, these viruses, and they grow them up in eggs. Yeah, chicken eggs. Because the virus needs some kind of cell to reproduce in, right? We've always said they need to reproduce in a host. Don't grow on a plate or in liquid media. So they're grown up, they infect the eggs, they reproduce in the eggs, they're killed. The viruses are totally killed, and then they extract the antigens. So the flu viruses are dead, right? You cannot get the flu from getting a flu vaccine, a dead flu vaccine shot, because they're dead. They're not going to come to life when they inject you. So that's usually how it's done. Now, there is now an attenuated vaccine option. You know, attenuated, that means they're greatly weakened, they're harmless, but they're not dead. And it's literally given as kind of a nasal spray vaccine. It's pretty popular with kids and people who are afraid of shots. Right? Some people are deathly afraid of shots, so this is a good option. And being an attenuated, you think, oh, it's attenuated. That's going to give me B and cyto T memories, right? There's more antigen. It should be a better vaccine, right? Well, a few years ago, they released an attenuated flu vaccine, and it just didn't work. I know on paper it should have worked better, but it didn't. They pulled it off the market, and now they're putting it on again. I have not seen how successful it's been. So we'll just kind of wait and see. But there is that option, attenuated, which gives you both memory B and memory cyto T, and then you got your dead, which gives you the memory. But this one has been around longer, and at least this one right now is a more reliable one. Okay. However, whether you get the flu or you get a vaccine, long-term immunity, meaning being immune to it many, many years, is difficult because, and we knew this was coming, antigenic variation. The flu changes its antigens. It changes the things it expresses over time. That's why you have to get a flu shot every year. Why one this year isn't good for many, many years. Because the virus is changing its antigens over time, which makes the memory B cells not as good as you'd hope them to be. Okay, so what kind of changes are we looking at? What kind of antigenic variations are we looking at? There's two kinds. The first one is what's called antigenic shift. And that's because, let's say the genes in the flu virus are undergoing point mutations. Like think missense mutations. Minor changes. Think missense mutations. So that's one amino acid change here, one amino acid change there. They're minor substitutions in the viral proteins. Minor substitutions. So you'd say, okay, who's making the mistake? Well, it's got to be the viral RNA polymerase, right? and it's making the mistakes during biosynthesis, right? So when the new RNA is being made, all right, when the new viral RNA is being made, okay, and that's going to be, remember, translated, okay, RNA is translated, and unfortunately there's mistakes in it. Mistakes end up in the protein. Those are antigens. So there are changes, therefore, in the proteins. There's changes in the antigens, and that's during the biosynthesis step, right? The third step. Now, here's when we have to do a little hand waving, and there's a gray area. There's no clear answer. The memory cells you have, they're partially useful, because in this case you'd say, well, there's several genes in a flu virus, and if they all change right away? Well, not really, probably not right away, and maybe some of the changes are pretty minor, so you'd say the antigens could stay the same, or at least they're similar. There are changes. So you'd say, well, the memory cells, they're not great, but they're still somewhat useful, because let's say the B memory cells, they get activated, they make plasma Bs, and those antibodies are decent, not great, but they're decent. They help your body, but they're not as efficient as they used to be when they were a perfect match for the antigen. So I just take a quote from one of the books I read, because you may get, so let's say you've had the flu vaccine, or you had the flu last year, and now you get the flu again. Okay, well, why? If you have the memories, because you'll get a clinical, okay, you'll get symptoms, but it's less serious than normal, because the plasma Bs and the cyto Ts from the memory cells work to some degree. So I'm just going to leave it to some degree. It's better than not having memory cells at all. Those memory cells are partially useful. They'll lead to partially useful plasma Bs that make antibodies and partially useful cyto Ts, assuming you had, let's say, the attenuated vaccine, or you actually had the disease, because you'd have both then. So I have to leave it in that kind of gray area. It's better than nothing, but it's not perfect. Let's go now to the second type of change, not antigenic drift. Now we're going to talk about antigenic shift. Okay? What does it have to do? Well, think an antigenic shift, think, oh my God, it's really bad. I mean, you feel like, look at the pallor. You feel like you're going to die. Also, antigenic shift is going to have to do with killing pigs and killing chickens even. All right, so what's going on here? What's the connection? Well, in antigenic shift, okay, get this clear. Don't get them confused on a test. Think shift. Shift is a big change. Shift is like you go in a car. You're going to go from park to drive. Shift is a big difference. If you drift, like we just talked about before, minor, small changes, you drift in a car, you're still moving, but you're just kind of changing just ever so slightly. So shift is a big change. Drift is a minor one. So we talk antigenic shift. What are we talking about? Well, it starts with this idea. Okay, first of all, if you have a avian flu virus, a flu virus that infects a chicken, it normally cannot infect a human cell. And a human flu virus, a flu virus that affects a human being, usually it cannot infect an avian cell. So human flu viruses, the ones that attack your body, aren't going to infect a chicken, and vice versa, right? So you'd say that the viruses are host specific for the most part. However, here's interesting, that an avian flu virus, one that usually infects chickens, and a human flu virus, one that usually infects people, can both infect a pig cell. They can infect, Porky. Porky the pig. Yeah, they can infect that. Why is that important? Well, let's hear it. So we'd say there's a co-infection of an avian and a human influenza viruses, both kinds, into one pig cell. So you go, okay, so what? Well, that's the basis of antigenic shift. Both viruses will reproduce inside a pig cell. And remember, viruses, all the parts are made, and then they're assembled together, right? And then they're sent out. Okay, like mixing up a salad, tossing a salad. Well, sometimes new viruses form because they have human virus parts and avian virus parts. Or we just say they have avian virus genes and therefore avian virus proteins and human virus proteins that are tossed together. So they're put together, but you're getting a mixture of parts. So we'd say that's a mistake during, not biosynthesis, during assembly or maturation, right? Assembly or maturation stage. So that's different than antigenic drift, which was a biosynthesis mistake. Okay, so you're saying, all right, how can we have a picture, Jeff? Absolutely. So here's the idea, first of all. Here we have a human influenza virus right here. Okay? And it's H and N. Okay, H and N. H and N on the surface. And there's genes for this human influenza virus. And that can infect a human being. All right? However, like I said, cannot infect the duck. Same token, here is a avian flu virus. Here's its H and N's, H and N's, okay? And it can infect a duck, perfectly fine, but it cannot infect a human being. Nope, can't infect that. But both viruses, okay, the human flu virus and the avian flu virus, once they're reproduced in a human, the human flu virus, or the chicken, the duck, the avian flu virus, those viruses can co-infect a pig. So it's going to go here. They go and infect the pig. And you can imagine you get a hybrid virus. And that hybrid virus, here's the hybrid virus. Look, it's got parts avian flu virus proteins, and it's got human flu virus proteins. It's got a mixture. Why? Because it's got a mixture of genes. You see that? So you got a mixture of genes and therefore a mixture of proteins. Now this hybrid virus can, if it's got the right combination, in this one we're saying it's the right combination, it can now infect a human being and potentially lead to a pandemic. All right? Because it's got the right proteins to infect, let's say on the surface, to infect the human cells. All right? So here's our example. Let's say you have an H3N2 human. All right? That's H3N2. Here's H3N2. Here's your H5N1. Here's your H5N1. Okay? And you put them there, you put them together, and inside the pig cell, you end up with an H5, H from the avian, N2, N2 from the human. It's a hybrid. And that one, now that combination allows that new virus to infect a human being, and it could be a pandemic strain. That's how there have been some flu pandemics in history, and that's how they got out. Antigenic shift. Bad. This one just kind of compares the two. If you just want to look at this, here's antigenic drift. Here's one virus. It comes in, mutations in the gene occur during replication, during biosynthesis. Okay? There's just some mistakes. It comes out. It's slightly different. Missense mutations. Antigenic drift. But in shift, you got two. 1 and 2. We're talking about a pig cell. A human flu virus and avian flu virus. They come in here, they're reproducing, and look, you got the different parts, and guess what? They get mixed together. These are all mixed together, all right? They're all mixed up, like tossed like a salad. Ooh, mix them all up. And they all come together, and guess what? They're going to go into this new hybrid one. Different genes, different proteins, but now it has the abilities, it has traits of both of the viruses. And the key thing is they can infect human beings. All right. So, and again, this is why these are severe. severe when you have antigenic shift. Mixing in a pig, it's called. Mixing in a pig. Now, what's so bad about it? Okay, there's two concerns. Number one, the hybrid virus has potentially the worst virulent genes of an avian virus with some human virus structural genes. And those human virus structural genes allow it to attack a human cell. So I say you kind of got, in this case, you've got the worst of both worlds. I mean, we all know Hannah Montana. You got the best of both worlds. Da-da-da-da-da. Yeah, okay. That's why I didn't make it as a singer. That's why I'm a biology professor. But now you got the worst of both worlds. Okay, so number one, why is it the worst of both worlds? Well, one is usually avian flu viruses are bad. I mean, they're usually very virulent. They really can kill a lot of birds. So they have severe effects. So you can think of the genes that encode the proteins for these avian flu viruses are bad. They're very virulent, high virulence. But now, so that's one thing. But now you've got them packaged inside viruses that can infect human cells. So think the H and the N combination allows the virus to now infect a human cell. So you've got the worst of both worlds. You've got really bad virulence genes, ones that were originally just in the avian flu viruses, and now you've repackaged them so now they can go into human cells because you've got the structural genes usually that help them get those viruses attack human cells, infect human cells. The worst of both worlds. The second thing about is this, what is also about bat shift. Well, the second thing is the antigens that are in this new hybrid virus, the shifted virus, are very different. Those proteins are very different from the ones that were in the original human flu virus. So you can imagine this new virus has very different proteins, has very different antigens, and therefore the memory cells that you've made against all these human viruses in the past, purely human, become useless, pretty much useless. Why? Because these antigens that are in these hybrid viruses express them, they haven't been in humans before. Those proteins, those antigens, they've been produced by avian flu virus. They've been in chickens and ducks, but they've never been in humans. So 3, 4, 10, 20 years, you've never have memory cells that recognize those antigens. These are totally new antigens. So you have a much weaker response than an antigenic drift, since again, these are totally brand new antigens and the memory cells you don't have any memory cells that not even kind of recognize those. This is what happened in the Spanish flu pandemic of about 1919. It killed 40 million people. Well, why? Well, one reason, was, you had the worst of both worlds. And secondly, people were getting this virus and it was totally new antigens. The memory cells were useless. So millions died. That's why. Okay. So now you're like, okay, do you think you understand then why you kill pigs then if there's a big flu epidemic? When you're worried about antigenic shift? Well, why? Because those new hybrid viruses, the ones that can attack humans that have the genes and make the proteins of the avian flu viruses, where are they coming out of? They're coming out of the pigs. The pigs are those reservoirs for those new viruses. All right, those new viruses aren't coming out of chickens. They're not coming out of people. They're coming out of the pigs because they were mixed in a pig. That's how you got them. They were mixed in a pig. That's when they're worried about this. They'll say, "Kill the pigs! Kill the pigs! Kill the pigs!" Right, and along with the chickens too. All right, I hope that makes sense. So let's go to a question now, comparing antigenic shift and drift. You were just infected with last year's flu virus, which has undergone antigenic drift since last year. Thus, you believe, you can pause it now and think about it. but I won't. Okay. Um, so it's drift. So let's think about the word drift. Minor changes. You're drifting in a car, slow. You're thinking missense mutations. You want to associate that with biosynthesis errors. The viral RNA polymerase just making mistakes when it's copying the viral RNA, right? So it's not mixing in a pig. It's not hybrid. It's not stuff like that. So what's true of that? The virus, no, no, not pig, no. The came about error in the assembly. No, drift is drift is not there. Ah, drift. Yeah, there you go. That's drift. Your memory, no, your memory cells will be somewhat good because the antigen doesn't change, but they're pretty small changes. So the answer would be C. Okay, everyone good? C. Now, if I had changed this one to the word shift, make one sure it has undergone antigenic shift, what would be true? Yeah, if it's shift, then A would be true, B would be true, and D would be true. Okay, those three, A, B, and D. Okay, put a little star. A, B, and D. Those are antigenic shift. Okay, C is drift. So just kind of keep those straight in your mind. Okay. So now we're going to talk about cold. And a lot of times people will tell me, "Oh, Jeff, I had the worst flu." I go, "Really?" I go, "Yeah, I just kind of had a really throbbing headache and my nose was stuffy for a couple days, but now I'm fine." And I'm like, "No, no, no, no. You didn't have flu. You had a cold. You had a cold, okay?" So, what's the differences? And colds are much milder. Very simple, very much milder. Like, but we'll go through them. But for instance, cold, rarely do you have a fever. Flu, big time fever. Um, Aches and pains, horrible fatigue and tiredness, exhaustion. Those are flu. I mean, flu is like, oh, I can't get out of bed for a couple days. Cold, yeah, you're kind of annoying, but it's not that bad. Okay. Um, but if you're really runny, stuffy, full nose and sneezing, that's much more a cold than a flu. Okay. Alrighty. And we'll like not going to go in here. And complications, that's going to be big too. We're going to talk about those later. So kind of knocking things off big time here on big that nothing. And these are all big time here, big time here, big time here, big time here. Okay? So let's kind of go, why are there changes and why are there these differences? Excuse me. So the common cold, again, if you want to read more about it, a lot of people are really into colds. They're giving you quite a few resources. So the agent of the common cold, there's many different ones. There's adenoviruses. There are even some coronaviruses, milder coronaviruses than COVID-19. But greater than 50% of colds are caused by this group called the rhinoviruses. Rhinoviruses. And there are over hundreds of types of these rhinoviruses. I think the latest thing I read was 167 or something like that of rhinoviruses. And rhino, what do you think? You think of rhino? Think of the animal. Rhino has to do with nose. So I think it's a virus that likes to infect the nose, and that's true. So these are sense-stranded RNA viruses. So flu is a antisense. This is a sense one. Okay. So you think, okay, do you need really quick? Do you need to have viral RNA polymerase as a soluble protein? And the answer is no. If they're sense, they can be translated right away, right? A ribosome can jump on them and translate them right away. They have no envelope, so there's going to be a lysis, okay? And I'll tell you, there's major lysis, major inflammation. So if cells are lysing rapidly, you can see a lot of inflammation. See that? They are very mutagenic because they have an error-prone viral RNA polymerase, kind of like flu. That's a true. Now, I want to make this clear. There's no antigenic shift with common cold virus, with rhinoviruses. They don't co-infect and then mix up their parts and come out with high. There's no antigenic shift. You can call this mutation, you can call it antigenic drift for rhinoviruses, but usually the word antigenic drift is usually reserved for just flu viruses. Even though I have seen people use the word with rhinoviruses, antigenic drift. But shift is definitely a flu thing, not a rhinovirus thing. Okay? Just want to clear that up. When you look at the rhinoviruses, what's most prominent is their surface proteins. And there's a model of it. Okay, there's your model. And it's got four major surface proteins. And they're called viral protein 1. Okay, that's I know, real creative. Rhinovirus scientists. Viral protein 1, viral protein 2, viral protein 3, viral protein 4. Viral protein 1 is the most important because that is the ligand. Okay, that's the ligand. So it's sticking out. And where's it sticking out? Sticking out of? Do you want to think about, well, it's a naked virus, right? It's naked. It's a naked virus. It's naked. So if you think it's a naked, then if it's got a naked virus, where's the ligand? It's going to be on the capsid. Very good. Capsid. Okay, capsid. And that's kind of, again, if you know your basics of viruses, these things are easy to predict. A little bit of rhinoviruses, they like to cool out. We say they're colds because they like to chill out in the nose. All right. Why? Well, they don't do well at 37 degrees Celsius in your body core. Okay. It's too hot for them. They're not made to grow there. They like to grow at about 33, maybe 34 degrees Celsius. Cooler. That's why they like your nose. And a little bit your throat too. Okay, a little bit your throat too. I mean, that's kind of, you're breathing in, it's a little cooler. But your core, which is about 37 degrees Celsius, you just can't do it. That's why rhinoviruses do not infect the lungs. They don't infect the lungs. Okay? They chill out in the nose because they're cold viruses. All right. Love this chicken soup. So for years, people have been saying, well, you should take chicken soup for a cold, chicken soup for a cold. Well, people think that it's not the vitamins or the nutrition. Some people think that when you're eating hot soup, okay, it's hot and the vapors are going up and you're breathing it into your nose, that's going to raise the temperature of your nose and that will somehow slow down rhinovirus replication. That's the story. But I've never seen any data to support that, really. Doing chicken soup under people's noses. Maybe some people, maybe somewhere at Stanford, they'll do that research. Who knows? Other things about the virus, they like being cool. They're very sensitive to detergents and pHs of less than 5. So like acidic conditions. They don't grow well there. So that's why you've seen these Kleenex. It's out by Kimberly Clark. They're known in the industry as killer Kleenex because they're actually contain in them a detergent, all right, in the clean, and it's slightly a lower pH, not enough to burn your fingers, but a lower pH, and that's why they say specially treated middle layers killed cold and flu viruses. Well, why the cold virus? Because the detergent and the acid. Okay, that's why. And so if you blow your nose into them, it'll kill them. Versus you blow in your hand or a handkerchief or just a paper towel or regular Kleenex, it's not going to kill them. Now, a lot of people think, well, is that good for me? I go, no, it's not good for. I mean, if you use these, basically, you're not helping yourself. You're already infected. You have the cold, but it stops you from infecting others. Because if you blow into your hand or a handkerchief or something else, those viruses are alive and they can be picked up. All right, so it's a good thing to help others, and you should do that because two reasons. Number one, you should care about the health of others. You shouldn't just focus on yourself. And number two, this is really important. I own stock in Kimberly Clark. All right, and I'm telling you, this has not been that big of a seller. And I got to pay for my retirement. So think about buying this, if not for yourself and for your friends, for me, okay? Come on.
So, how do you get the cold? How is it transmitted? Well, it can be by inhalation. You breathe in somebody's respiratory droplets after they cough or sneeze by you. But actually, it's mainly by contact, meaning someone coughs or sneezes or touches something, the viruses are on a fomite, for instance, and then you pick that up. You pick it up, you touch it onto your fingers and hands, and then you rub it onto your eyes or nose. So it's mainly through contact. So that's why people are always like, oh, wash your hands a lot, if you want to prevent colds. Because you can get it through inhalation, but mainly it's through contact, picking things up. All right? Keep your hands away from your face. The incubation time is usual, about 3-day rule. And here's an interesting is how long you for contagious for. All right? And you get a cold, and after the incubation period, okay, you start becoming symptomatic. First you get that raw throat, you start a little bit sniffly. And then after a few days, maybe that goes away, but then you get really congested, maybe 5, 6, 7 days into symptomy. Well, it's only the first 2 to 3 days when you're symptomatic, just the beginning when you start feeling it. That's when you're mainly contagious. Why? Because like I said, the virus is going to give up pretty quickly in your body. Your body's going to beat it pretty quickly. So by 5 or 6 days into being symptomatic, when you might be more congested and you can hear it in someone's voice, actually the virus is gone. You're no longer shedding. It's just your body's immune system is cleaning things up. All right. The symptoms and signs, these, dry, scratchy, red throat, raw throat, red runny nose, cough. A big thing about colds is there's usually no fever. That's a big difference from the flu. The flu has a rapid onset of fever, flu fever. But usually most colds, especially those caused by rhinoviruses, which we're focusing on, rhinoviral colds, there's no fever. Okay. So let's go on.
So we kind of look at our thing. We're kind of revisiting the chart, and we're kind of saying, okay, let's kind of we're kind of trying to understand. We've kind of got the fever thing. Um, we kind of got the nose and the throat. Okay, we've kind of talked a little bit about that. All right, inflammation. Think bad inflammation. Okay. All right, but how about and we've kind of talked before about the flu, why you get the fever there, and we've talked about the fatigue. Remember, cytokines like interferons and general aches and pains and exhaustion. So we're kind of getting there, but let's just kind of keep moving on. So in the pathogenic mechanism, what's going on? What's the ligand? What's the receptor? So VP1 is the ligand, and it binds the receptors that's on your throat, pharyngeal cells, also nose cells. Sorry, put nose in there. And it enters by what? Okay, the ligand binds a receptor. Remember, it's a naked virus, so it's got to enter by direct penetration. Okay, so it enters by direct penetration. Now, here's a really interesting soluble protein that's found in rhinoviruses. And it has a very fancy name with Greek letters and stuff like that. But on my tests, we're just going to call the beer goggles protein. Beer goggles, the world's worst invention. And you're like, what? And you might have heard beer goggles. You might have heard the saying, you go to a party, you're sober, you look around, no one's particularly attractive to you physically. You drink a few beers, and then they say, now you got on the beer goggles. It affects your vision, your perception of people. And suddenly maybe someone who wasn't that attractive because of the beer looks really good. So they say, well, you're wearing the beer goggles. The beer has made someone attractive to you. Well, there's a protein, fancy scientific name. We don't care about that. We're going to just call it the beer goggles protein. And it is a soluble protein that's in the nucleocapsid of rhinoviruses. And you're like, well, why is it necessary? So we'll think about it. The rhinovirus enters, it has one RNA strand. So and it's like mRNA because it's sense RNA. It's like one mRNA. So it's kind of like Waldo in this crowd. And I can't find Waldo, but let's say Waldo's here. So there's one viral mRNA that's entered the cell. It's a sense strand. It's like an mRNA. And what else? Well, in the cell, there's all these human mRNAs. Everybody else here, everybody else is a human mRNA. Everybody else is a human mRNA except here, Waldo. That one there, okay? So why would ribosomes, I ask you, why would ribosomes go after the one viral mRNA instead of all these human mRNAs? Well, there must be something that makes the ribosome attracted to the viral mRNA, to the viral genome. What is that? The beer goggles protein. The beer goggles protein, it's a viral protein, it's a soluble protein. It's got to be there. It's a soluble. It's one that's used right away. And it comes in and it goes to the ribosomes and it makes the ribosomes want to go after that one viral mRNA and not instead of going after human mRNAs. So you can put in the word human mRNA. Pause. Okay. So after that, then the virus, okay, then it's the virus is going to have its mRNA translated. There'll be viral proteins, and then you're going to get a lot of viral proteins and more viral mRNA, and they assemble, and now you've got all these things going on. Okay, just think about sense-stranded viruses. Okay, so now you get a lot of new rhinoviruses made. Now, as the virus reproduces, and remember, it's going to leave, and it's going to leave by lysis. We're going to see a lot of inflammation. Okay, cell lysis triggers a lot of inflammation. All right, well, what do you think with inflammation? Histamines, leukotrienes, right? And your body overreacts, okay, because there's too many leukotrienes. So what you get all these things of inflammation, red, sneezing, congestion, all that fluid, right? The inflammation, it's blocked up. Inflammation, pain. All right? So that's how you're seeing most of the symptoms. They're from that really bad inflammation resulting from this lysis of cells. Okay? Of the cells. Now, also you're like, well, why doesn't the body stop it real quick? Well, the viruses decrease your non-specific immune response. One of your major ones to cold rhinoviruses is interferons. And that's what's really interesting. They have a very interesting way of basically inhibiting the production of interferon by your body cells. So when a body cell gets infected by a rhinovirus, it wants to send interferons, remember, unselfish gifts to other cells to warn them, but a rhinovirus stops interferon production. So that's one of its little strategies to stick around in your body for a little bit and fight your body and take on your body and reproduce in your body. But not totally. Why? Because your body basically, between its macrophages and complement and other parts of the innate immunity, it will pretty much get rid of, with macrophages, complement, it'll pretty much get rid of most of the rhinoviruses. B cells and cyto T cells help, but they take a while to come on, and usually the rhinoviruses are pretty much been eliminated by your body by the time you get a lot of antibody and cyto T response. It's why, because rhinoviruses are like Italian soldiers. They give up pretty easily. All right, I always say Italians, we're not great fighters. I mean, we switched sides in World War I and World War II. You didn't know that? Yeah, a little history. But we're great lovers. That's what we say. And so rhinoviruses give up very easily. They get knocked out pretty easily. So at that point, there's really not that much for the antibodies and the cyto Ts to do. But, as they say, those who fight and run away live to fight another day. So So secondly, well, what happens though if you do have a bad cold? You do get inflammation, you get destruction of cells. So it's worth noting that if you get secondary infections, the damage has been done in the upper respiratory tract, right? Nose and throat, not the lungs, not the bronchioles and the trachea and the ciliate. No, not in the ciliated respiratory cells. It's done up above. So what are the bacteria that attack up above, okay? In the upper respiratory tract. So think of Streptococcus pyogenes, strep throat. So that's why strep throat often is a secondary disease of a cold, of rhinovirus. Okay? Pneumonia usually is not. Usually. Okay? So that's why. So that kind of should explain the differences. A flu leading to pneumococcal pneumonia, a cold leading to things like strep throat. Now, what can you do if you get a common cold? Well, there's no cure. Basically, all you're going to do is treat the symptoms. Number one, antihistamines. Basically, to nullify the effect of histamine. Remember, histamine causes inflammation, so you want to decrease the inflammation, in the nasal passages mainly. Secondly, you have decongestants. Decongestants decrease nasal secretion. Well, why? Well, because they also trigger vasoconstriction. They are also anti-inflammatory. And slightly different mechanism. The antihistamines, same result. To just treat the aches and pains you have, you take a non-steroidal anti-inflammatory drug such as Motrin. Okay, it's just to help symptoms. And finally, what most people tell you, just drink plenty of fluids because you lose a lot, because you're sneezing stuff out, stuff like that, and get plenty of rest, and that will kind of speed up your recovery time, so your body will overcome the cold and get back to normal. All right, and feeling good. Yeah, there's a lot of like homeopathic stuff out there, medicine, family tradition, like drinking brandy in a big tub of hot water, or echinacea, or airborne. But there's only really scant evidence that those things help. The best thing you can do right now is just treat the symptoms and get plenty of fluid and rest, and hopefully you recover quickly. All right. Let's get going with part two here. So how about prevention or perhaps a cure for a common cold? Well, you'd think a vaccine would be a perfect thing to do, right? Here's a little cocktail. For a school age kid, grammar school, on average they can contract about 12 common colds a year. Wow, 12. Now it kind of goes down as you become an adult, but you can just see so many people get sick. Vaccine would be great, right, to prevent all this. Well, there's two main reasons there's no vaccine for rhinovirus, for the common cold. Number one, there's so many different viruses, so many different types of rhinovirus that can cause the common cold. All right, and that doesn't even factor into account other kind of viruses like adenoviruses that can cause the common cold. It'd be really hard to, for instance, if you were going to do a subunit vaccine, to put enough antigen of all those different strains, something like 150 or so of those rhinovirus strains. It would be very hard to get enough of those antigens to actually do anything into one or two shots. Just too many viruses involved. Couldn't represent them all, okay, in a vaccine. Couldn't get all the shots there. And secondly, rhinoviruses are very mutagenic. Remember, the viral RNA polymerase makes mistakes, all right? It's mutagenic. That means the antigens change when you go over several generations. So the memory cells that would be good for one type of strain would not be good later in time because that strain has changed its antigens. All right, so that's kind of the two key reasons. And finally, like I said, there's a lot of home remedy cures, but which I just talked about a little while ago before, but there's no actual scientific evidence that supports any of these cures, whether it be echinacea, zinc, other things you might have heard of right now. All right, but the search goes on, and hopefully one day they will figure out actually how to make a vaccine. Maybe they'll have a new strategy, or they'll come up with some type of pharmaceutical that will actually be good against common cold rhinoviruses. Okay. Now, the last thing about colds is how to prevent them, because no one likes getting a cold. What's the best thing you can do? And I know that a lot of people say, oh, take vitamin C, take hot bath, take echinacea. There's a lot of stories out there. But really, the best documented thing you can do to prevent getting a cold, an infection by rhinovirus, is what you've heard before for COVID-19: wash your hands. Okay? Keep your hands away from your face. The idea is you don't want the virus from somehow getting to your respiratory tract. All right? Because, like I said, yeah, you can get it by inhalation of droplets, but it's often by contact. People sneezing somewhere, you then unwittingly touching that area, picking up those viruses, and then touching your eyes, your nose, things like that. Okay? So wash your hands. Soap and water, 20 seconds. The second thing you can do to prevent colds is to sleep enough. And I'm just going to try to illustrate this idea with an experiment that was done just about 4 years ago at UCSF. And as you can see here, sleep protects against the common cold. So they took several groups of people and they basically ate pretty much similar food, drank things, did similar activities. Everything was fairly controlled. The only thing they varied among the four groups was the amount of sleep they got per night. So one group they got over 7 hours. One group got between 6 and 7 hours of sleep. Another group got between 5 and 6, and there was the last group that got less than 5 hours of sleep. All right. And what they did for all of these people was they literally infected them, or potentially infected them, with the same number of rhinoviruses. What occurred was they drew up a same number of rhinoviruses into a pipette, and they injected it down people's noses. So it would go into their noses and then in their respiratory tract. So all these people in all the four groups got the same number of rhinoviruses into their respiratory tract. Everything was pretty much the same among them. What they ate, their activities, everything. Again, with the exception was how much sleep they got. Now, who came down? What percent of each group came down with the clinical signs and symptoms of a cold? Who got sick? Well, the people that got more than 7 hours of sleep, 17% came down with a cold, signs and symptoms of cold. Pretty amazing, isn't it? Only 17%. Even though everyone had gotten the quote unquote, what was considered to be the dose. The group that got a little less sleep, 22.7% came down with a cold. A little bit more, not much. 5 to 6 hours of sleep, 30%. Wow, that's almost twice as much as those that got 7 hours of sleep, greater than 7 hours. And finally, if you got less than 5 hours of sleep, the chances are 45.2% of those people, they came down with a cold. So wow, it's like almost from 17 to 45, almost 3 times as likely to come down with the signs and symptoms of a cold if you got less than 5 hours of sleep versus if you got a decent night's sleep of greater than 7 hours. So you can see the importance of good sleep to preventing you from coming down with a cold, even if you are exposed to what's quote unquote considered a dose of cold viruses, rhinoviruses. So that's why, folks, when I get emails from some of you and you write them to me at like 2, 3 in the morning, I think to myself, uh-oh, those people are really running great risk of coming down with a cold. Try to get more sleep, folks, please. Okay, the last viral respiratory disease is one you've heard a lot about. I think it's COVID-19. And COVID-19, by the way, stands for coronavirus vi disease of 2019. So that's how you got the name. The agent is a member of the family of coronaviruses, and that family is known as Coronaviridae. And the actual name of, the virus is, some people will say the COVID-19 virus, but the formal name is SARS-CoV-2. Okay. Now, the coronavirus is a very large family of viruses. All right. And there's a lot of members that cause pretty mild colds. Okay. Common colds. So it's very likely that you've gotten a cold that's been caused by other members of the coronavirus family. However, SARS-CoV-2 is one that causes the most virulent kind of disease. It's known as corona because corona is Spanish for crown. And if you look at the surface of the virus, you see the spikes, right? The spikes. And the spikes give it a crown-like appearance. Okay. Now, it is an enveloped sense RNA virus. Now, you know the basics of viral life cycles. So just from that, you know already a lot about SARS-CoV-2. Okay, now it is enveloped, so you say, well, it could enter by fusion or endocytosis. I'm going to tell you it's going to enter by endocytosis. All right. If it's entering by endocytosis, then you know about the uncoating, right? It's going to require the acidification to melt away the 3 layers, right? Which we talked about before. See, you know the basic, you can apply it real simple. Just looking here at its entrance, here you have SARS-CoV-2. There's the spike. Okay, the spike has got the ligands. Okay, it's got the ligand. And it's going to bind to receptor. And you've learned from Lab 8, the receptor is ACE2. So human cells that express ACE2 on their surfaces, they're the ones that are going to get infected by SARS-CoV-2. If they don't have it, they don't get infected. So what happens is the spike protein ligands bind the ACE2 receptors and it gets endocytosed. Now, how does it start? Well, not surprising. It's mainly transmitted by inhalation of respiratory droplets. And it was also believed is by contact, by taking those droplets if they came off a fomite and touching your face. But as you read in one of your recent required readings, they're not so sure about that. They know it's definitely inhalation of the actual droplets when they're in the air, but how long it can last outside the body, how long it can last on a fomite is not clear, and they don't really have many cases that have been verified showing that it's from contact. But nevertheless, you can pick up a lot of things from your hands, so wash your hands nevertheless. Okay? Good. So once you've picked it up, it's been transmitted to you, I'm just going to go quickly over the highlights of the life cycle, which you can predict from your basic knowledge of its structure. Number 1, the ligand binds the receptor, ACE2. It is endocytosed. Okay, all right. I know it says memory, but remember it's the acid is pumped in, which allows for its release. Okay. The mRNA, it's a sense strand, so it can be translated right away by the ribosomes. So it's translated, and you get these, remember polyproteins are made? They're fusion proteins, and they need to be cleaved then by proteases. All right, so now the proteins, here's your viral proteins, excuse me. And those can be like the viral RNA polymerase, more proteases, capsid proteins. You get the idea. All right, now remember, you've got to make new RNAs for not only to be more translation, more protein made, but remember, you need more RNAs to be going into the baby viruses, so genomes. So what's really important is the viral RNA polymerase does transcription from the RNA, makes more RNAs. Okay, it can inhibit it by some nucleotide analog inhibitors. We'll talk about those. All right, but basically these are base analogs. So that's used for treatment of SARS-CoV-2. Once the RNAs are made and the proteins have been made, they assemble. The formation, they assemble. All right, and then they're going to bud off. And now these viruses can go infect new cells. Right?
So let's talk a little bit about SARS-CoV-2 by the numbers. The burst time is 10 hours, and the burst size is 1,000. So imagine one virus enters a respiratory cell. In 10 hours, that one has become 1,000. And then if those 1,000 all go into cells, another 10 hours later, 1,000 times 1,000 would be a million. So 20 hours after infection, with just one virus, you get more than one at a time, of course. But if you just had one enter your body, you'd have a million times that number. So pretty impressive. The diameter of the virus, interesting, knows about 100 nanometers. That's a zero point 0.1 micron. So, and you might have heard they're always saying you should use N95 masks. And why? Because N95 masks are capable of about filtering almost 100% of particles that have a diameter of about 0.1 micron. That's the diameter of SARS-CoV-2. So it makes sense. That's why people are encouraged to use N95 masks if possible. Kind of interesting point. The genome, like I said, it's a virus, doesn't carry a lot of genes, travels light, has about 10 genes. Now, interestingly, the virus has 26 functional proteins. So you usually think, well, one gene, one protein. How can that be? Well, think back. Viruses, remember, sometimes the proteins they make are polyproteins or fusion proteins, meaning that one protein actually gets cut up, hint, by a protease. So that one fusion protein is cut up and it actually has maybe 2 or 3 or 4 actual proteins in it. So one gene codes one mRNA. It's translated into one big, long polyprotein, but the protease snips it up into several proteins. So that's how 10 genes can encode for 26 functional proteins for SARS-CoV-2. Lastly, people have always asked me, well, where do you find the viruses? And number one, of course, the respiratory tract and the sputum, that's the stuff like deep in the lungs. And also, you'll find it in the nasopharynx, right? The nose and throat, and that's usually where they do the swabbing, all right? Because it's kind of hard to reach down into the lungs. So usually if you've got them down there in the sputum, they'll also be in your nose and throat, and that's where they swab it to test you. And even they found in some poop, there's virus in poop. Now, luckily, the viruses in poop are not very contagious. There's something about them that doesn't make them very contagious. So that's why they're usually spread just by respiratory droplets. Okay. So how contagious is COVID? The incubation time is about 5 days, all right, on average. However, sometimes depending on the person and the strain, it can be 2 to 9 days, all right. And in general, you are contagious during the incubation time, all right. The basic reproduction number are for the original COVID virus, which came out in 2019, was about 3. And like I said, the infectious time, the time that you're actually infectious, contagious, about 7 to 14 days between during incubation time and when you're actually showing symptomy. The R value, like I said, for the first one was about 3. We've got these newer strains, let's say the Omicron ones. The R is guesstimated, approximated to be even more than 10. So again, that's one person can give it to 10 people, alrighty, assuming they're not vaccinated or they have had previously, don't have any immunity from before. So it's a very contagious disease, as all of you know by now. So I'm going to get into symptomy in a second, but I want to talk a little bit about long COVID here. Most people will get over the symptomatic part of the disease within 2 weeks. So about 2 weeks of being symptomatic. By the end of that, most people are done. But there's still some people who get what's called long COVID. And it's estimated maybe about 10% get long COVID. And that means their symptoms can last to months to years. All right. And at first, a lot of people thought, oh, these people they're just talk. They just want to stay home from work longer, But they've shown that there is scientific proof behind this. People do get long COVID. And it's not clear why. They're still researching it. It's a big mystery. Now, when you're in the long COVID period, okay, so after that first 2 weeks, you feel these symptoms still, but you're not contagious. All right, seemingly not contagious. So like, well, what's going on? Well, a person suffers like brain fog. They have muscle tightness, muscle aches. Maybe they have that nagging cough. It really depends. Depending on the person, you have slightly different symptoms of long COVID. What it has been associated with, though, is number one, persistent inflammation. You person still has inflammation in different parts of the body, which you don't expect. All right, it should be gone, but they do see that correlation. Sometimes inflammation in the heart, inflammation in the muscles, blood vessels, things like that. They'll see inflammation around there. And secondly, they see autoantibodies. So you should remember that from we talked about rheumatic fever. You have antibodies. Your body's made antibodies against that attacks its own parts of the body. So long COVID is very interesting, and it's something that they have not figured out yet. Okay, let's just get to the symptoms and signs of just regular COVID, not long COVID. Estimate, first of all, is like can be up to 50% of people that get infected with the virus have a subclinical disease. All right, so they never get sick. And it's kind of hard to figure this out, in studies, but that's what's estimated. Now, they're contagious, but they're asymptomatic reservoirs. So they don't suffer it, but they can pass it on to others. There was a really interesting study done in a small city in Italy called Vo, Italy, years ago. It was near the beginning of the pandemic. It was kind of a isolated town, and they can kind of like control things and study things because not many people went in, not many people went out. And they found that, again, it was a large percent of the people, they were saying that actually got infected, but they were asymptomatic and they never came down with it. And there were some people, of course, that, a little bit more than the majority, more than 50, they actually, they got infected and they became symptomatic. But when that first came out, a lot of people would not believe that. But it's found out true that a lot of people about COVID stay asymptomatic. However, that's why, quote unquote, healthy people, if they've been exposed, they should wear masks around immunocompromised people. Because you can say, well, I'm healthy, I'm fine, but you could be one of these asymptomatic carriers. And as I'll explain in a second, if you're immunocompromised and you get COVID, it can get really, really bad. So about 80% of the people that are symptomatic, they have mild to moderate symptoms, cough, fever, shortness of breath, very similar to a real bad cold, all right, plus the fever thrown in there, all right, but they get over it, all right, mild to moderate symptoms. But what was so worrisome about it is that about 20% of those are symptomatic. They develop really serious symptoms that usually lead to hospitalization, all right, where they have like problems of having, for instance, they have insufficient oxygen in the blood, all right, they can possibly die, all right, very serious. So they need hospitalization. Usually these are people that are badly immunocompromised, like the elderly, okay, people that are suffering other diseases, people have a lot of health complications, diabetes, really bad obesity, things like that. But some, for instance, that got really, really sick are completely healthy on the superficial level. There are people that don't have any obvious immunocompromise. And you probably read about people like that, all right, or know people like that happened to. So it's just a very scary disease in that way. So let's kind of look at the pathogenic mechanism here, and let's kind of focus on people with the more severe symptoms, the ones who end up going to the hospital. So it mainly begins, as in all people, it mainly affects the throat cells, upper respiratory tract, all right? But if you have some immunocompromise or for something, it can then progress further down. And this is when it gets bad because it'll hit the ciliated respiratory cells. Okay, you've heard about that. All right. And those are, of course, the cells, trachea, bronchi, those are the mucosal escalator cells. And in certain people, when you get the severe symptoms, they'll go down into the type 2 pneumocytes. Those are the cells in the alveoli. Okay. So once they get to the alveoli, you know that the bloodstream is very close by. So the viruses can actually get into the blood. So you'll see viremia. All right? All right, so now the viruses can get to other parts of the body. Right? And like I mentioned a little while ago, you can see like heart inflammation and things like that. And it's thought because the virus can get to the heart through the blood and things like that. All right, so that's why it kind of hooks into some of the long COVID symptoms and things like that. Secondly, with it going down into the mucosal escalator and damaging that, if you damage the mucosal escalator, now you can have secondary diseases, bacterial pneumonia, which we talked about before. So that's why people will say, oh yeah, they have COVID, but they develop bacterial pneumonia. Well, that's why. Okay. Um, with this, you have obviously compromised lung function as the viruses, work these cells to death, kill them off. All right. So there's not going to get enough oxygen to the body. All right. That's a problem. All right. That's why people often put on respirators. In this scenario, there's a lot of inflammation about COVID. COVID triggers the body to do like way too much inflammation. So there's a lot of pus buildup in the lungs, things like that. So obviously the lungs are not working very, very well. And they're like, well, how about shock? And yeah, shock's a possibility. So as kind of shown in the little drawing below here, what you can do is suffer from a cytokine storm. So literally a lot of like immune cells in your body will end up releasing a lot of extra levels of cytokines. So it's not it's like a super antigen, but it's like the same effect that lots of different immune cells overproduce these cytokines. These are cellular messengers. And what they'll do is in the body to lead to, again, excessive inflammation. All right. And such that there's a lot of inflammation. Imagine fluid leaving the bloodstream into the tissues. Well, you're going to have less blood volume. All right, so that if there's less blood volume, number one, oxygen doesn't get to all the cells in the body, and number two, less blood volume, less blood pressure. You've heard this before, shock. Okay, there are a lot of players in this inflammatory response, in this cytokine storm. However, probably the major one that they've kind of focused on is called interleukin-6, IL-6. You want to kind of put that aside in your mind, IL-6, and they've shown like sometimes some treatments for people who have really bad COVID, they'll give them IL-6 antibodies to neutralize IL-6. So IL-6 is something your body makes in normal, moderate amounts. It's good, but something about COVID infections, way overproduction of IL-6. IL-6 is a cytokine, and overproduction of that and other cytokines, cytokine storm, and you see all these problems. All right. So let's talk about treatment and prevention of COVID-19. First of all, for treatment, there's a drug been around for a long time. It's called dexamethasone. It's mainly to treat the symptoms and the signs. It's a very good anti-inflammatory. And as I've talked about before, inflammation is one of the major problems when you get COVID-19. Secondly, to actually try to decrease the duration of the disease to stop the virus life cycle, there is one thing that they use in the U.S. called remdesivir. It's a base analog, so you get shortened RNA genomes. Secondly, there's molnupiravir. That causes a lot of mistakes by the viral RNA polymerase. They call them like catastrophic errors, big mistakes in transcription, so that the RNA genome that's made has a lot of mistakes, and therefore that causes the virus to die off. The third and probably the most common treatment for COVID-19, which you've probably heard about because they advertise it a lot, is Paxlovid. And Paxlovid is a protease inhibitor. So it's going to basically stop the polyproteins or fusion proteins from being properly cut up and activated. All right. Now, the next thing is you might have heard of people saying, oh, take hydroxychloroquine, take hydroxychloroquine. Well, it's been proven by quite a few scientific studies it's not effective. Now, why they thought it was going to be effective is because they know that hydroxychloroquine changes the pHs of endosomes. And like I said, this is an envelope virus that comes in by endocytosis. There's going to be endosomes formed, and the acid that gets pumped in is necessary for uncoating. So theoretically, hydroxychloroquine should work. But in reality, for some reason, it's not clear to me, it doesn't work. It does not. It's not effective at stopping the virus going through its life cycle. For prevention, there are several approved vaccines, thank goodness. The most common ones are from Pfizer and Moderna. Now, they are effective for some people, somewhat effective in preventing you from getting sick, preventing you from getting the clinical infection, the signs and the symptoms. Okay? But for a lot of people, you get the vaccine, you still get the illness, but it's a lot milder signs and symptoms. Okay. Well, it's not perfect. But what it's important, and really the key thing about getting the vaccine, is it's almost 100% effective in preventing deaths. Okay, that's the key thing. The vaccine, true, might not be able to prevent you from getting sick, but if you do get sick, there will be milder symptoms and signs, and pretty much sure it's going to prevent you from dying, and that's the key goal. Practically, and you know this, for what can you do practically to prevent the spread of COVID? Wear masks, socially distance, and wash your hands a lot. Okay. Final point of the day. What's the state of COVID right now? Well, things are a lot better than about 6 years ago when the pandemic hit and they closed down everything and everyone had to go online. I think everyone remembers back then. Life's gone back to pretty much normal in terms of travel and stuff like that. Uh, could it get bad again? Yes, it could get bad again. I mean, you never know. The strain is constantly mutating, and you hear about new strains coming out all the time. So far, symptoms change slightly, but they're not that much more virulent. But there's always a chance that one virus comes about that has a really bad combination of genes, and it can be much more higher in terms of mortality. But hopefully it won't. I think the best way is to be prepared, get vaccinated, keep up with your vaccines, and just be positive and hope that doesn't get worse. All right, well, I'll leave it at that. You folks lived through the pandemic. You can tell your children and your grandchildren about it, and it gives you a very unique perspective. So take care, be safe, and I'll see you in lecture. Thanks so much for listening. Bye-bye.